Thank you. And I have a lot of cables attached. It doesn't mean I'm a cyborg. It's just your cables are attached here. So let me, I have to playing to catch, and I'm hoping the auditorium here knows a little bit what NGS is and what high performance computing is. So I'll be talking quickly about Hive itself, and then I'll dedicate about 5 minutes on what the biocompute paradigm is and what we at FDA are trying to promote, the transparency of the computational universe. So in Hive, and Hive, like Lawrence told, it stands for High Performance Integrated Virtual Environment. The abbreviation became up later. The reason it's called Hive because it's always buzzing. There's a lot of computations going on. So challenges, quickly. What does it represent from FDA perspective? I'm pretty sure you know about all of this. These are pathogens, vaccines, viruses, clinical markers, outbreaks, food safety information, etc. Who produces it? Everybody produces it. Industry, academia, you produce it, we produce it to understand that, evaluate, and create new methods of evaluation of a regulatory medical products. And why do we produce it? Because it's so cheap. I don't know if you know this fact, but the fastest growing data is the NGS data at this point. Not Hadron Collider, not physics, not atmospherics, nothing else. It's the NGS is so cheap to produce. For $100,000, you can buy a high-performance sequencing machine in a lab, and you can start cranking the data. If you want a satellite, it costs you $1 billion to launch it. So NGS is just the beginning of an NGS, and we produce a lot of it. And we analyze it because we hope there is a treasury of information inside. And why do we keep it is very important for two reasons. One of them is that we don't trust our current understanding. Every day, new algorithms are being published. Every day, new level of interpretation is appearing. And we at FDA also have a mandate to some point maintain an information which has been submitted to us for regulatory evidence. We have to maintain that information for longer than research scientists usually should. So data is big, and data is being generated very fast, and the variety of data is big. Now, just when I started this, there was a DNA sequencing and whole genome shotgun sequencing. Now we have RNA-seq, immune-seq, TN-seq. There are many seq, and then it's just the sequencing part of it. Once you move to the big data universe, you start getting non-sequencing projects into it. And there's a humongous data analytics field, and we get all kind of data. Variety is big. And when we are talking about the regulation, it's also very important for us to understand where the data appeared. How did you analyze how did you prepare your sample? How did you prepare the libraries? How did you sequence? What was the bioinformatics pipeline? Just the amount of information is not enough for us. We have to have all of the metadata and understanding the generation of the outcome. So classical sequencing pipeline, we get a sample, we run it through sequencing machine, we transfer it to archival, then we start taking it from archival system, we compute on it, and someone very smart and intelligent looks at the data and then makes a regulation. We have a bunch of problems. We have files are humongous, moving it is very difficult, and transfer is slow. Sometimes to transfer the information, it takes us weeks to months to go through all of the security checkpoints to get the data in. Privacy is a big issue because some of that data is proprietary and some of that is research. Maintaining it is very difficult. Validation of the bioinformatics pipeline is difficult. 85% of all analytics on an NGS space is spent on alignments, and then about 10% is a variant calling and coverage analysis, and there are about 100 different alignments algorithms you can use. So validating if your program is better than the other ones, or even if it's valid scientifically, is very difficult. So let's move forward. So we need a platform which helps us to do some of it. So that's why Hive has been designed. Let's move to another kind of an analysis, nothing to do with NGS, a consolidated metadata analysis, let's say from post-market space. We get data from different spaces, then we have to do ontology mapping. That's to say like, hey, your fields are called ABC, my fields are called differently. How do we know what value in your tables corresponds to what value in my tables? Because they are coming from different databases. Sometimes it's sex, sometimes it's gender, sometimes it's male, female, sometimes it's M and F. What are the units? What are the dosages? Are they normalized, calibrated? All of that thing have to be done. And then how do you store this data? How do you access the data? Is it just you are storing the PDFs, and then how do you query the data? How do you analyze that? So there are problems. Again, I'm not going to go to details of the problems. Size is a problem. Ontological mapping is a problem. So we need a platform which helps us to do that too. So these are two clear justifications. Why does FDA need a big platform which is very capable on not just getting the data, but getting it safely, securely, and being able to analyze it? Okay. So the old days, let's go back to the sequencing. Old days of sequencing, you would get few sequences, you'd log into an CBI website, for example, click a couple of buttons, copy paste your sequence, go drink a coffee, come back, the data is ready. Today, that doesn't work like that. There's too much data. It will take you days to upload to NCBI, if NCBI lets you do that. If you click on a button and go, it will never finish, and you will find out it didn't finish only in hours. So it doesn't work. So there are challenges. I'm not going to go to details. Data is a challenge, size is challenge, archival is challenge, standards are challenge, and then complexity of the data. It's not just one blob and you have all metadata companies to it. You know how what describes what. Computation is the same thing. Very difficult because it's big. I've computed some of the computations we do on a daily basis. It will take 56 years to do in your notebook. How useful that is. So obviously we need these humongous compute clusters. So the old days of doing science when you have the data and you have the good brain and you want to analyze are gone. Because now you have to have powerful computers. Oh my God, now you have to build a network and host the computers, go through purchase, acquisition, etc., etc. So just to do the basic science, now today we have to deal with a big infrastructure. So let's talk about that infrastructure, the platform, Hive platform. It's an encapsulated data platform which has an archival system and compute system integrated under a common, behind the common firewall. Yeah, I use that word way too many times. So there is a common web portal, and there is an API-driven website which allows you to get the information, load the information. We can directly go and fetch the information from your devices which are connected to the same network. We can go and fetch the data from the external data providers, let's say sequencing the centers where you send your samples to, or from your local hard drives. Everything is web-driven and web browsable. So you can come to Hive and command Hive to go and fetch the information, and we'll do it for you in a parallel fashion. Once, let's say you go to SRA, you have an SRA session numbers or GenBank identifiers, you tell us, we get the data for you, and we hold it in a distributed fashion in this big and humongous cluster we have built. So when you need to do the computation, you come to the system, you select your data sets which are already in a system, few buttons down the road, and the Hive will take your computation request, understand how complex it is, how big it is, split into thousands of pieces and send to the compute cluster. Monitor itself that every single chunk of a computation is done, orchestrate where the computation are going based on where the data is. Sometimes it will move the data to the computation, sometimes it will move the computation to the data. And if jobs fail, and believe me, jobs fail. I mean, when your Word crashes once in a while, because you just loaded a big file, it's okay, you just restart it. When you are running millions of computation a day, things crash and you see them much more frequently because you are running a million computation a day. So who does the restarting? Who glues the pieces? So that's what Hive is. Hive is a big orchestrator platform. So another big issue which we are facing is the number of data types to describe. You know, when you hire a few DB administrators to build a platform for database for you, and then they design 20, 30 databases, now you have your data type. The next day scientists come and say, well, I need another data type. I need another database. So soon you recognize you need an army of DB administrators working for you just to design data types and ontological mapping. So that doesn't work. So inside we created this honeycomb database this infrastructure which pretty much allows the scientists to design the data types and populate it themselves. I can go into many details. Unfortunately, time will not allow me to do. Deployments. This platform is, it started like, it's one of these before and after pictures where it happened. So we started from a bunch of cables and computers connected on our desktops. Now today, there are multiple deployments which look nice like the one on the bottom. So we have a Maxi Hive. It's a regulatory analytics platform, which is about 2 petabytes data storage, 3,000 compute cores. And then we have a Mini Hive, which is a research production platform, again at FDA. These two are inside FDA. We do not mix and match the data because regular data live in a separate space, research data lives in a separate space. Again, it's a powerful platform. It has 1,500 cores. So we have a public instance of Hive. This is for public consumption. It's not as big, but it still is very powerful. And that is for people who want to collaborate, who want to develop new algorithms, or just use Hive for their own purposes. And then we have public elastic Hive. That's again in Ashburn. It's a big data center where we are. So you can, if you need more resources than the one which the public Hive provides, you can do it, but then you pay the electricity bill. So we have, how big is Hive and how small is Hive? So we have about 400 different data types, which is worth about 20-30 different databases information. During the last 3 years, we have run 1,500 years of computation, and the cluster is under constant 80 to 100% load. So one of these days when I was showing this slide, somebody said, hey, but this is not optimal. We have a green cluster. Our computers, when we are not using, we are shutting down, and you are constantly proud of Hive buzzing all around. So, and then we have a green cluster. Why don't you follow the technology and build a green cluster? I don't know how can you build a green cluster if you're 80 to 100% load all of the time. Why would you spend the money and buy computers and then be proud of shutting them down when you don't use them? So I think we are really proud that all of the hardware we put our money in is being strictly used. And one of the reasons is because we don't force our users to code and develop. It's all web-driven, and it's all easy. So we have also Amazon instance of running. Within 15 minutes, I can bring a cluster up. In another 2 hours, I can have the cluster actually full of data, like reference data materials, human genomes, annotations, NTs of the world, and then you can have an instance working there. So again, people sometimes ask, why would you even do Hive platform development if there are all of these clusters and SunGrid engines? Yes, they are, but they are just a bunch of hardware. We need, who does the coding? We do the coding. We build the infrastructure for how you bring the data. So Hive, when we built at FDA, not as yet another hardware platform or software platform, it's an end-to-end solution. It pretty much helps you from the point to get the data to the point when you are actually publishing a publication or doing a regulation. Because the scientists, and it's easy because scientists should not be doing a coding. These people, scientists who we work for, spend tens of years actually perfecting their scientific skills. Why would you force them to go under the tables connecting the clusters or running the C, C++ codes or things like this? Because when you talk about Python, Perl, Java, C, and Shell, for scientists, those are the ones on the left. But when we are talking about these languages, these are the coding languages actually. The ones we mean is on the right side of the code. So, and this is how the actually scientists look like when they see the code, and this is how I look like on the bottom. So I actually had a case when I was showing one of the slides, somebody said, oh, that sounds very great, 1,000 cores, what is a core? So yeah, we don't make scientists to do non-science, they all do science. Okay, so a lot of time when we are presenting, people are coming back to me and asking questions, then tell us the story, how did you build the cluster, how big it can get, because we have so much data, we want to make a big cluster. So you have to choose your tools right. I mean, today, when we joined FDA, this is where we were. We needed a small thing to solve a couple of small questions. But then we recognize tomorrow we are going to be here. So we are not going to choose a hammer, we need a crane. But we also recognize in the future we are going to be here. So it's very important when you are building your system to choose the right tools. Don't solve the small questions if the next one is much bigger. And then there is we recognize something. When we started building the cluster and growing, we recognized there are limits to how much you can grow. So in the real world, mammals have a limitation on size. They cannot be too small because they will lose heat and die. And they cannot be too big because they'll cook themselves, because there's the amount of heat which is generated by the mammal is proportional to the cube of its size, but the amount of heat it can dissipate is the square of its size. So obviously this is a clear example that there are limits of viability of the mammal species. Similarly, because we love our computers so much, like our pets, we recognize there might be some principles affecting them too. So you can't just grow the compute cluster and hope it's actually doing a good job. Because once you increase it too much, there are limits. So just for mammals, this is a balance between heat. Let's say a simple example of heat and the resource demand and resource consumption and dissipation. Similarly, computers have the same issue. You have to bring the storage up, and we have to cabling cost, and then heat dissipation, heat generation, and development cost. All together, there are limits how big you can grow. And the problem is very simple. Let's say I have 10 nodes of hardware, okay, 2 nodes of hardware storage, and 3 nodes of the computation. That is a perfect cluster. There is no latency on waiting the data. As soon as the computers come up, they have the data ready. But when you increase the cluster size twice, the amount of interaction goes as the square of m, because they're randomly accessing the data. That means you need 4 times bigger amount of networking. So this is not sustainable. The problem is that you cannot just grow your cluster and be happy, and it's not going to do more. At some point, you are increasing the size of the cluster, size of the high-performance computing system, it slows down because there's so much interaction and intertalk that you are spending the money to make the thing worse. So you cannot just throw the money to it, you have to be smart to it, and you have to design it well. So at some point, we recognize that just increasing the size of a cluster doesn't work, so we are doing this multi-Hive-led fractal expansion, that we are growing each instance of a Hive to the point, and then we are treating the democratizing the system, like there's instance of Hive there, instance of Hive there. Data doesn't move, computations move if necessary, do the computation and only outcomes move. So let's go further. Hive is a data retrieval system, it's a storage system, security system, it does Tailor meta-analytics. Okay, let's go forward. Because we are FDA, a few more slides about the security of it. So once you have too many security rules, systems slow down. Systems can slow down that 80% of the time you are wasting not on computation, but on actually making security checks. And when you work at FDA, you recognize that there's a regulatory data which has so many security credentials that are associated to it. So to avoid all of that, we designed this new hierarchical security system. We have a hierarchy of organization and projects, hierarchy of the objects and data. So the way we share the collaboratively data, that's a completely different platform, not existent in any Linux or Windows system that you are familiar with. I can go into details more. It's also data segregation and compute segregation. We have a way of controlling that. This is a regulatory computation. It cannot coexist in a parts of the system where research collaborations are happening. So security, another attribute here. So when we say sharing the data, it's not just the data you can share. In Hive, you can share your data, your computation process itself, the outcomes of the computation. You can share menu buttons or new algorithms. You upload a new algorithm, you share an algorithm. Also, so because of all of these security credentials we attach to it, now we got authorized to operate in a regulatory environment with a regulatory data set. Believe it or not, even if it's an FDA-developed system, it still needs to go through all of the compliances and things like that. So it's also FISMA moderate. That is to say that we can be trusted to be operational and safe and secure. And there are multiple layers of security. Okay, integrator. Okay, so let's say we build the platform. What can it do? So can I build a new software for it? Yes. What kind of hardware it can run? So it's agnostic on a hardware. You can run Linux, Windows, SunGrid engines. I can launch it in your single notebook. What operational systems? You name it, whatever ones are popular, right there. So languages, major languages for development, right there. So we can do tools natively develop. You can pretty much, there's a code base you develop based on it. Or you can bring your open source tool. It will be integrated. Or if it's a command line utility, I can wrap it into Hive application and launch it under Hive. The same way you get the performance of a Hive, but it's your tool, it's your existing system. It also can be RESTful API driven. So there is a web API. You can pretty much, from your computer, you can drive Hive to do something for you. So this is just a couple of slides in case I attached for demonstration, in case if the questions come. So integration, we are moving to integrate. We'll see other applications and other platforms are integrating currently. Because of the regulatory compliance of Hive, there is an interest to integrate with it. So CLC Bio, you might know, is being integrated. Galaxy is being integrated. We are in a very close collaboration with NCBI, and some of the algorithms are moving both ways. So we have Hive apps, and also we are at the core of FDA's attempt to standardize the NGS world, and we are integrating with post-market analytics, and we are running the collaborations across all of the centers of FDA. So this is about integration. So let's talk about the software. You recognize when you work 3.5 years on a project and develop every day, you cannot present in few slides what you have done. Yes, this is just to mention a few. We work in NGS space. We have all of the aligners, variant callers, RNA-seq, in silico tools, comparative genomic annotations. There are about 200 different applications developed inside Hive, and about like 40, 50 adopted popular applications. So this is going to be a big system. And everything is visual. Our users never see lines of code. All they see is these nice, beautiful visualizations. That actually helps them to do a lot of work. Think about the performance. Why do you need a performance? You might say it's big computation anyway. And the reason is very simple. When you are a scientist, you never know what question is right to ask because you have to work with the data. You have to ask few wrong questions before you know what the right question is even. So it's very highly performing system. Things which run outside for half a day to 2 days can run in minutes in Hive. And there is no magic. It's just a bunch of computers we have connected together and made it easy for you. So So, and then once the outcome, how useful it is if I say, I give you your result, 300 million row table, please take it, are you happy? No, of course not, because you can't even look at it. So we had to develop for almost every application separate visualization technique. And they're all very interactive. You touch them, and every touch and click brings up 100 computers behind the scene who have to take the look at these 300 million tables, prepare an excerpt of the data, prepare a simulation, a visualization based on it, and then show it to you. And just once a person asked, well, I waited for 15 minutes for the human genome alignment. Now I have to, after every click, wait for 5 seconds before you show me the zoom of a human genome. How about that? 3 billion human positions, 200 things we know about 200 bytes of information about every position. That's a 600 gigabyte information right there. You click a button, within 5 seconds, I am supposed to show you a beautiful picture, what you are expecting. So behind every visualization, there is a large amount of computation and work to be done. Just to give you samples, you understand there are more than 80 projects running in Hive. This is the way we look at the metagenomic samples. So this is a sample which has been accumulated from next to a restroom in a farm. And then you load it into Hive, and then you click few buttons. In 10-15 minutes, you get what kind of genes, what kind of genomes have been hit. So in this case, we have found disease agents, gut biome, plants, funguses. We have found HIV, chlamydia, dogs, and horses, and everything in 15 minutes. Because that's a farm, and all of them are going, and everyone contributed into that pool of water. Another sample is, ask a question, please.
First of all, thank you. I've already learned an enormous amount, but I have 3 questions.
Sure.
One, you mentioned an API. Is there an API available for
Yes.
Hive?
Yes.
And if so, through the API, can one actually program in this environment, or is this really intended to be a GUI type?
No, you can. I mean, you can. If the tools you are trying to drive exist in Hive, you can use those tools to drive your analysis.
What sort of tools you probably went over this and I missed it, but
so
like a high level of what kind of computer?
Yeah, so because we started as an NGS platform, obviously we have NGS aligners, variant callers, comparative genomics, and things like this.
The thing that you showed like 2 slides ago.
Yes.
Top hat and
Yes. So some of the tools, and behind everyone there are multiple algorithms, multiple implementations, etc. Yes.
And what about data structures?
So you can data structures as per metadata information, or?
No, I mean actually. So you've provided essentially all the data schemas and all the data structures.
Yeah. Of course, there's the data structures behind them.
And you've settled on that. Okay.
Yeah.
So that's it. And then, is there a plan to basically distribute this architecture and software as an image or as a solution to regional centers?
So we are going to go open source. It's all waiting our public announcements and things like this, and we are clearing the code. Because we are FDA, it's very cumbersome. You have to go security process, clean the code from all of the comments, all of the things before we can go.
I was just wondering if that was something.
Yep. Yeah, it's on a plan. It's going to go open source.
Do you think if that's available that people are going to be able to run that in virtualized cloud centers?
Yes, you can run in cloud center. Performance might be suffering because, let's say you're running a cluster from Amazon cheats and puts your computers in a virtual nodes where other things are running. They're not cheating. I mean, that's the way it's designed, virtual. So you are getting a performance hit, but you will get it running.
And then finally, I mean, is this the basis of a sort of a regulatory environment in which you expect NGS analysis to occur?
So we are doing, and Eric might talk about this, we have run a regulatory analysis on this. Yep.
So, I mean, meaning if someone is actually developing something that's intended to maybe be some sort of new tool.
Okay, I'll touch that. I'll touch base that. I'll touch base that biocomputing. Yep. Yes?
Just one quick question. On your metagenomics, is that whole shotgun sequencing or is it 16S RNA?
No, it's a whole shotgun sequencing in this case. So it's not 16S. 16S, it will take much lesser time to do that. This is 15 minutes, whole 15 minutes to wait. I know it's fast. I am proud to say that. Yeah, we worked about a long time to come to this performance level. Yes. So another sample is that we deal a lot of viruses and vaccines and things like that. Yes. So classical understanding of viruses as a piece of a chromosome from left to right, that at some point stops working for us because of people call it quasi-speciation. I don't like that word quasi-species. It's just the population diversity of the viruses is humongous. So let's say you are studying exactly that. You have a sample of the virus from a patient or from a stool sample or from an environmental sample. So within few minutes, as soon as you load into Hive, click few buttons, this computation, the top one takes only 10 minutes. We will assemble the clones. In this case, you can see 2 clones in a polio vaccine sample. Every trajectory from left to right is a separate virus. So you can see this region of the virus is different. There are 2 clones here. The left region is different, and the right region are identical. This is a conserved region, and these are distinguished regions. Yes, they are different enough to be assembled clones. So this is like kind of a de novo assembly on a viral metagenomic population. So the middle sample, I think, is that stool sample. Again, you can see more clones forming here. It's again polio. And the bottom sample is actually sewage from Israel, which we got. You can see hundreds of clones are forming there. Each one has a distinct pattern of mutations from every other one. So again, I can tell about this tool for hours, how it works, how good or bad it is, and compare it to others. It's just a sample of like what information you can get. No, we were not able to even mimic the same level of computation on any existing tool. So another one, and this is a work with one of my very good collaborators, Malcolm Moose, who is actually Stanford. His alma mater is Stanford also. So we are doing single-cell RNA-seq, and on a fluidigm chip, they are running mesenchymal stem cells and separate cells inside of the fluidigm, and then RNA-seq protocol is run, library is prepared, and we measure about 200,000 RNA splice variant expression in all of these cells, separate cells. Then we collapse it into genetic space, so we pretty much have an expression profile of hundreds of samples, tens of thousands of the expressions. So what do you do with that? Yes. So now your NGS is finished. We could just wash our hands, say it's not our job, but hey, we need to finish the analysis. So, and that is an ill-defined, mathematically ill-defined space. So we had to drop NGS part of the work, move it to the actually data science analysis, and develop classifiers for them in a 20,000 dimensions. Let me tell you this, there are no tools to do that which are publicly available. So we had to completely redesign ours in a very high performance computing system. So in this case, you can see that it's a 3rd and 7th day oxygen stress of mesenchymal stem cells. If you look at this data when it's unclustered, so pretty much it looks like a big mess. You don't know which RNAs are playing a role during the differentiation of the mesenchymal stem cells. Once you run through this analysis, FuncDA analysis, it actually takes only a few seconds to run a 10,000 dimensional linear discriminant in this case. So we pretty much come up, within few seconds, you come up with the coefficients which you can stick into parfait analysis and see which genes are expressed or underexpressed, or which linear combination is responsible for phenotype separation. Again, I can talk hours about this. We don't have time. Well, I mean, I think we are time limited, so I'll just jump. Those are just samplers that the outcomes are actually computation visualizations. But in Hive, we never develop things for no reason. So we only develop around a particular goal. That's why all of our tools are tailor-made for a purpose. So every time somebody comes and say, do you have this? We might have 80% of it, 90% of it. But because it's always tailor-made, we usually need another 10% to assemble our tools differently to make it useful for our scientists. You know, this is a classical example. Stradivarius was never assembling a violin by putting a string into the violin. He would actually attach a string, make a sound, bring the piece of wood to it, see how the sound changes. So he would assemble the wood around the string, not the other way around. That's why violins from Stradivarius are not that good, even so many years. But again, when you have a good tool, you a good tool is just a tool. It's just like a scalpel. Yes, you can do something very useful, or you can do some damage with it. Yes, so with every good tool, there is an expertise to go, and that is very important. And that's what also we try to provide in Hive for FDA, that level of expertise, because once you have too much data, you can come to any conclusion you want. You have to be very careful in the way you use the tool. So let me quickly go on a biocompute concept. One of the important tasks we are trying to play is help FDA and validate the bioinformatics pipelines and understand how can industry communicate bioinformatics to us. The days of measuring intensities of the lights and in a big table and doing some basic statistics to it are gone when the next gen is here. So complex pipelines are being employed, and way too many times we get results of analysis, we don't know how it came to be. I'm going to jump a few slides here. So I'll be back. So when an industry submits something and then to FDA, after they did their data analysis, let's say data forming and computation, and submit to us, so we sometimes can reanalyze the data inside our system and come up to regulatory decision. It's good when everything works together and everything agrees, but frequently they might not. So then there's regulatory loops are happening. So we communicate back the information, and they do something with the data, again communicate back to us. And regulatory loops cost money, and that is very expensive. Few weeks cost millions of dollars. So we want to change the schema by making it more transparent the way we do our computations. So we are developing this biocompute paradigm, which pretty much is very simple. It says, hey, these are the JSON objects, and where you have to define what are your protocols, what are the pipelines, how do you put internal parameters, what are the parameterization, and send it to us. And whatever we are using, we are going to put outside all of the SOPs. We are working currently to put all of the standard operating procedures of NGS analysis from Hive to public consumption. Next year, March, there is going to be a conference where this is going to be announced to the world. And GW is working with us. So the idea is now the companies or anybody, academia or anybody, can take our protocols and run these analyses in an integrated platform outside of FDA. You can do as many iterations as you want to, and then you can submit the data. But because you did analysis, now you know what we are going to see. So if you don't agree with us, you can still submit the data with an explanation why the differences are coming here. Or maybe we can see something, you can see something in your data while you are running our analysis also. This does not mean any analysis is better or the other one. It just means it is a transparent way. How do we look at the data? How do we look at the submitter's data? So, and then to do biocompute, very simple. We look at what is the actual experiments are happening. You always have generalist experiment. You have inputs, you have parameters, and you have outputs, just like in chemical experiment, except that experiment itself is the application running behind or a pipeline. The inputs are your input samples files, and the parameters are a bunch of command line arguments, and the outputs are some tables. So let's go forward. So at every point when you're running an analysis, a good analytical pipeline may be perfect under certain parameterization for some kind of analysis and be absolutely horrible for other kinds of analysis. And we have seen these cases when somebody takes the human variation calling algorithm and does virus analysis and sends us the research data for us to look at it in a research collaboration. So this cannot be done, obviously, and the reason is that because they never looked how it was parameterized. So what we are trying to do in biocompute, you have to communicate what is the usability domain. How can I use this application? What is it for viruses? Is it for bacteria? What is parametric domain? And what are the knowledge domain? Because some algorithms can produce 20 different things. Some are tribal, some are trustable, some are not. So also, what is the knowledge domain and what is the range of errors? Again, the good tool, I already mentioned, can have multiple validity domains, yes? Under certain parameterization, bowtie, sum tools pipeline is good for humans. Under different parameterization, it's good for viruses and bacteria also. It's an example. So we are developing this platform where there is going to be public data typing engine. I think you mentioned, question was that what are the structures? So pretty much anybody in the world should be able to actually design new data types for their own computation using the public portal. And then based on that data typing engine, they can define data types.
So you said distributed computing quite a few times, and there are a number of available tools for that. I mean, can you tell us a little bit more?
Distributed?
distributed computing. Did you write your own, or are you using
Yep.
an available solution like Hadoop or something?
No, we didn't use. We actually tried Hadoop or others. Performance was not enough for us because it's running a Java core and we are dealing in next gen. A lot of time when you're running a next gen, even every single step of a transformation of the data, bytes moving, wrapped into the variants of the Java or something, they're too expensive. So we actually, Hive itself is, the core of Hive is developed by itself from scratch. I was a part of the NCBI long time and I was a contractor there. I developed high performance computing cores. Even today, PubChem or a third of the services at NCBI are running on my code base. So when I moved to FDA, I pretty much shared the ownership with an FDA for a similar platform. We actually made it better here than it was at NCBI. So it's a separate code base developed from scratch.
In the biocompute environment, for people to participate in that, to be able to submit their bioinformatic workflows, is there documentation about that that's available?
So it's going to come up. The work is being conducted as we speak, and the March is going to be announcement, and there's going to be a big conference. You are welcome to participate. I think it's 16, 17 March, but there'll be federal registry if you follow, and all of these things are going to be there. There's going to be federal registry, and all of the input is obviously welcome. The whole point of that conference is to get people like you to talk to us, because we don't have an idea that we know everything. In fact, we know we don't know everything. That's why we need input. Yeah. So just for the sake of time, it's a hierarchical data models which we are developing. So instead of every time from scratch developing a new data type, you will be able to use one of the data types which is defined before you and then just enrich with more specificity. I know I have a time constraint in many cases, so I might stop there and then pass the estafette to Eric Donaldson. And I have my slides I can share with them, and these are just samples of biocompute objects which are going to be there. These are drafts. My slides will be able for you to actually share and analyze and communicate back to me. So please do welcome, we do welcome collaborations or interactions or any smart idea you have.